Good evening, everyone. I welcome you all in the clinical research class. As of now, we have discussed the basic introduction of a clinical research and the history of a clinical research. So, in last class, we have discussed we have started with the phases of clinical trial. So, we have discussed all five phases that we are having in the clinical research. The phase zero is the microdosing, where we are going to obtain the preliminary PK data, and phase one is the stage where we are going to have the healthy volunteers, and we are going to check the drug for the safety and the tolerability. Then we have the phase two. So phase two is also known as the therapeutic exploratory phase, where we are going to confirm the safety and the efficacy of a drug. Now this is the particular phase, or you can say the first phase, which are having the patients. Then we have the phase three. So phase three is also known as the therapeutic confirmatory trials, where we are going to confirm the results that we have gotten from the phase 2. Here we are going to establish the efficacy of a drug in larger number of patients so that we can collect the safety data. Then the fourth phase that we are having, the fourth phase that we are having is the post-marketing studies, or PMS, or post-marketing surveillance. So in this phase, what happens? We are having the NDAs, that is the new drug application. We can market the drug, and we are going to see the effects of a drug in the larger population where there is no fixed population, duration is not fixed, so that we can detect the ADRs, that is the adverse drug reactions, and also if they sometimes we get to know the explore the new use of a drug. So these all the phases that we are having in the clinical trials. Then the next thing that we have discussed is the history. So in history,
Ma'am,
yes, Anamika.
uh, ma'am, I have read somewhere that phase two is also divided into phase 2A and phase 2B.
Yes,
So,
yes, 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 yes. Yes, we are going to have a proper class for the phases. As of now, I'm only giving you the basic thing, but all phases, what basic things we are getting from the phases. So we are going to, in upcoming classes, we are going to have a proper class on the phases, where we are going to discuss all these phases in a in a great depth, so that you can have the idea about the all the phases. Then we have discussed the historical events. So in historical events, we have seen first there was a Avcina, who has done some experiments on his patients when he was doing his medicine practice. So in 1020, so he was the first person who has done the experiment. Then we have seen the James Lind. So James Lind was the Scottish surgeon who was deployed at the ship for the sailors, and there he has conducted a own kind of clinical trial, like we can say, like he has taken the patients, he has divided those sailors into a group, and given them different treatments, and see what treatment are having the what effects. So this was the own and the first own kind of clinical experiment that we can say. And on the occasion of the on this occasion of this particular event, we are celebrating World Clinical Trial Day on the 20th May every year. Then we have seen the in 1906 we have the US FDA. So Food and Drug Act was there. Initially it was enforced by the Bureau of Chemistry in the US department. Later on, which in 1930, its name was changed as the US FDA. And so we have discussed why the need of the FDA Food and Drug Act was there, because we have seen like there was adulteration in the food, there was misbranding. We have seen like the alcohol and afim was added as the chief ingredients for the children medicines, and also we have seen like there was a lot of not the good things, dangerous things, or adding preservatives, dangerous preservatives were added into the food products. So like formaldehyde was added. So all these things, we have some publications from the different journalists who have published all these issues, who has raised all these issues. On that basis, the government felt that we should have some rules and regulations for the for all these things, so that by having the Food and Drug Act, so it's also known as the Willis Act, Willis law. So by having the Food and Drug Act, we was having the we were having the laws for the to ban the foreign and the interstate traffic, and also the adulterated and mislabeled food. So it was the first time when these things are also penalized. Like we are going to put the penalties for the misbranding and the adulteration, and also we are going to like it's important to mention all the ingredients that the particular product is happening having. So all these things was there in the Food and Drug Act. Then after the Food and Drug Act, so the therapeutic goods legislation was there by the Australian government. So they have introduced their own legislation where they have defined the drugs, the therapeutic devices, the role. So the role for this particular the role for this particular legislation was to protect the human health and the safety of the population, and it was aimed to ensure the safety, quality, and the efficacy of the therapeutic goods. So, like Australian government has also told that there was like as per different agencies were having different countries were having their regulations. Like in some countries having the national level agencies. In some countries, we are having the like state level agencies. So in Australia, we are having the national level agencies. So like as per the agencies, there are rules and that needed to be applied. And then we have learned about the Medical Research Endowment Fund Act 1937. So this was the fund which was given by the Australian government to the medical researchers, to the universities for the research, to the institutes for the medical persons who are there for the training. So this was the fund, like kind of fund, so that the Australian government provides so that the person can conduct their research, or the institute or any like research institutions or universities if they want to conduct any research. So this fund was allocated to them. Then, like in 1947, like we have a there was subsequent Nuremberg trial. So there was in like after World War II. So Nazis has conducted some brutal experiments. So which led to the formation of the Nuremberg Code. So Nuremberg Code is like these are the set of ethical principles for the human experimentations created by the U.S. Code after the subsequent Nuremberg trial that held after the World War II. So these trials were started in the 20th November 1945, and we get the Nuremberg Code in the 1947. So Nuremberg Code has basically the 10 points. So those 10 points include that the ICF should be mandatory, the experiment should be fruitful, we should have a preclinical data prior conducting the research, no physical and mental suffering should be done to the subjects, no experiment should be conducted where there is a prior reason to believe that the death can occur. We should always evaluate the degree of the risk and the benefit. The proper preparation and the facility should be there for the conduction of a clinical trial. Then the clinical trial should be conducted by the scientifically qualified persons. The person should have a proper knowledge. Then the patient can withdraw at any time from the trial. And the last one is that scientists can terminate the experiment at any time. So if they feel like the experiment results are not good enough, so then they can terminate the trial. So these all was the 10 ethical principles that we have gotten from the Nuremberg's code. Then in 19 like after Nuremberg's code, so we have another tragedy that is known as the thalidomide tragedy. So in 1961, it was a tragedy that like in late 1950s and early 1960s, there was pregnant women who were suffering from the morning sickness, anxiety, and trouble sleeping, and they was given the thalidomide. And after taking the thalidomide, it was seen the babies, the hundred thousands of babies which were born, they were with the severe deformities such as phocomelia. So phocomelia was seen. The like deformity and malformation of arms and legs was there on a large level. So there was a FDA officer known as the Francis Kelsey, who was the first officer who denied to provide the approval to the thalidomide. And after the efforts of the Francis Kelsey, this thalidomide was banned, and we have the regulation for the regulating all these things. So like after this, we have the Declaration of the Helsinki. So in Declaration of the Helsinki, we have the set of ethical principles which was given by the WMO. It is the World Medical Organization in 1964. And this Declaration of Helsinki is also known as the core-stone document on the human research ethics. So by having Declaration of Helsinki, we have basic principles that are moreover similar with the Nuremberg Code. Some new things are added like the protocol reviews required from the IRBs. Then the state compliance should be there. State compliance means we should have the regulatory approvals from the regulatory authorities, and we should also require to safeguard the rights of the participants who are there in the research. Then the privacy should be preserved. So like sorry, not the privacy, the accuracy should be preserved when we are publishing the data. And also we have the four principles from the Declaration of Helsinki, that was the autonomy, beneficence, non-maleficence, and the justice. So these are the four principles that we have gotten from the DOH. Then in 1966, we have some human experimentations. So like Tageski syphilis experiment was there, which were done on the African males. So they were having the syphilis, but they were unaware of it, and they were used, and like they were not provided proper medical care. So this lead to the formation, like after having this experiment, Tageski syphilis experiment, in 1979, we have the Belmont report. So Belmont report is like the report where we are having the principles. So from the Belmont report, we have the principles like respect, beneficence, and justice. So like I will explain all these reports in upcoming classes. So as of now, I'm just correlating all these things so that you can understand like from where we got all these things. So Belmont report was created by the National Commission for the Protection of Human Subjects of Biomedical and Behavioral Research, and the three primary area on which they focus was the informed consent, the assessment of the risk and the benefits, and the selection of human subjects in a research. And also the three principles that they have from the Belmont report is the respect, beneficence, and justice. Then in 1989 we have the Therapeutics Good Act. It was from the Australian government, and the therapeutics good regulation. So these particular regulations, these are the set out given by the Australian government. So they set out the requirements for the inclusion of the therapeutic goods in the Australia, like how what will be the regulations for the therapeutic good in the Australia. So the registration of goods are required. Advertising, they have like clarify all the things on the advertising, labeling, and product appearance, and also the right of an individual that affect them, like the right safety well-being of an individual should be maintained while conducting the clinical research. Then in 1997 we have the ICH-GCP. So ICH-GCP is the International Council for the Harmonization Good Clinical Practice. So like by having the ICH-GCP, we are having the ethical guidelines, scientific principles that like as of now they are also in the rule. So we are all, as of now we are all using the ICH-GCP for the conduction of a clinical trials. So in ICH-GCP, basically we have the four guidelines. So first is quality guidelines, then the second one is the safety guideline, third one is the efficacy guideline, and fourth one is the multidisciplinary guideline. So the most important thing that we are going to use is the efficacy guideline. So in efficacy guidelines, we have a guideline that is known as the ICH-GCP E6. So it's an international ethical scientific quality standard for designing, conducting, recording, and reporting the trial. So ICH, like ICH E6 GCP, so it says that particular guidelines is the it's the international ethical and scientific quality standards that we are having for designing, conducting, recording, and reporting the trials. And we have 13 principles from the ICH GCP. So like these principles need to be maintained whenever we are performing any clinical trial. And then in 1999, we have the national statement by the Australian government. So that were contained, like they have the Australian Health Ethics Committee developed the guideline that were contained in the National Statement on Ethical Conduct in a Human Research. Then in 2007, we have the revised version of this. So National Statement Code of Research. So it was revised in 2018. So this was the joint venture that was from the National Health and Medical Research Council and the Australian Research Council and the Universities of Australia that provide the guidelines for the researchers, human research, and ethics committee, and also for the all other conducting ethical reviews. So these was the like the timeline for the history of in the history of a clinical research, so that we can know when we get particular guidelines, so that we can have the idea from which incidents we are having those guidelines. So anyone is having any doubt up to there?
No, ma'am.
Okay. So the next thing, like we have already discussed all these things. Let me just read out these slides so that you can have the idea what is all here in these slides. So clinical research and clinical trials is a fundamental tool of a modern medicine, as you are already aware. Clinical research and clinical trial is a medical invention that has contributed nearly all of the life-saving medicines that we know today. Because whenever we are going to have the clinical trials, then only we can have the approved drugs in the market and we can use those drugs. So like it's added 10 years of our life expectancy. Then the made organ transplants possible. It's easy for transplanting the organs. Made diabetes a manageable condition. So like we are having the drugs for the diabetes, for curing the diabetes. Extended the life expectancy of the AIDS patients. Like as of now, we are having like drugs for maximum diseases. So that is only possible due to the clinical research. Prolong the lives of million of the cancer patients. So these are the some good benefits that we are having from the clinical research. Then it's history. So like as we have seen, like there was some history that was not there in that. So in 500 BC there was some Egyptian documents and their prescriptions was there. So they were on that basis like they had provided like whenever they are providing some medical care to the patient. So what all they have documented and their prescriptions, but there was no proof that they have done some work. So this was the only document. Their prescription documents was there, but we don't have any proof for those conduction. Then in 600 BC, there is Daniel of Judah that has conducted the comparative trial. So what was the comparative trial? So they have done the comparison between the vegetarian diet and the royal Babylon diet on the patients and seen like what all differences were there. So this was one kind of comparative trial that they have done. Then in 1754, we have the James Lind who discovered that the citrus fruit prevent the scurvy by conducting the clinical trial on the sailors and conducted first controlled clinical trial. So this was the world's first controlled clinical trial. Then in 1976, the Edward Jenner conducted the famous trial that proved the inoculation could prevent the smallpox. So this was conducted by the Edward Jenner. So Jenner trial was the first step towards the mass vaccination that conquered the epidemics such as typhoid, yellow fever, and the polio and the measles. Then later on in the 1863, Austral field used the placebo for the first time in the clinical research. So placebo is like it's a non-active the placebo is it look like similar as the drug, but it does not have the like it does not act the active pharmaceutical ingredient. So it is like we are only having placebo so that we can see the psychological effects of the person. So whether they are it's help like psychological things are helpful for curing the particular indication or not. Then in 900, so like first we have the Food and Drug Act, which prohibited the interstate commerce and adulteration. Then in 1912, we have the first regulation against the false therapeutic claims. Like if they can't make any false therapeutic claims, so labeling should be there. So US product the labeling laws enacted. Whatever is there in the medicine, whatever is there in the product, it should be mentioned on the label. Then in 1924, the first use of randomization in a clinical trial was by Emerson in a pulmonary tuberculosis trial. So randomization is the process. So whenever we are having the two groups, so we want to divide the drug into the two groups. So we have a process that is known the randomization. So randomization is the process, the allocation of a drug to the different groups that we are having in a study. So it's a process of allocation of a drug to the different groups that we are having in the study. So nowadays we are having different softwares on which we are performing the randomization. Then in 1946 we have the British epidemiologist Sir Austin Bradford Hill put the patients into the experimental and control group at the randomly eliminated bias. So they have he has performed the trial into the experimental and control groups at the random eliminated by any bias. So like so that the biasness should not be there. So he has put the persons into the two group. One is the experimental group and other is at the control group. And also this was done by the use of randomization so that the biasness could not be there. So biasness is like whenever anyone is trying to manipulate the results, or a biasness could be like it can be uh, like biasness can be we can say that can be intentional biasness also, and it can sometimes it can be an unintentional biasness. So to eliminate that particular biasness we use the randomization, because we are not aware like which patient is there in which particular group, because we are using some process on which the allocation of the patients was there. So it's automatically reduce the biasness of the allocation of the patients to the particular or the particular interest of a group. Then in 1930, the Therapeutic Trials Committee was first body to manage the clinical trials in the UK. So anyone is aware which is the for the US we have the regulatory body as the US FDA. For India, what is the regulatory body? CDSCO. It's CDSCO.
CD.
Yes. CDSCO. That is the Central Drug Standard Control Organization. For UK, is anyone aware what is the regulatory body for the UK? It's MHRA. But it is MHRA. So for UK, it's MHRA. So they have the regulatory body that is MHRA, that is the Medicines and Healthcare Products Regulatory Agency. Okay. So then in 1938, we have the first regulation for the requirement of the drug safety database for the US FDA, that is the Drug and Cosmetic Acts. So first regulation was there. So first multi-centric center studies employing the same protocol and thus enabling the pool of data to increase the statistical power. So in 1938, we have the revision of the Food and Drug Cosmetic Act was also done. So we are also having the safety database. Safety database is the database where we are going to have the information about the adverse drug reactions and the adverse events. Then in 1900s, we have the Nuremberg Code. So I have already explained the Nuremberg Code. Then in 1962, we have the Kafauver-Harris drug amendment. So I think we have not having this thing in our that history timeline of the history. So what is the Kafauver-Harris Amendment Act? So it's also known as the Drug Efficacy Amendment. So it was amendment to the Food and Drug Cosmetic Act. It was the amendment to the FDA Act 1938. So the act that we are having in the 1938, it was a kind of amendment to that. So it's basically introduced the regulation of providing the proof of the effectiveness and the safety prior the approvals. Like nowadays, whenever we are going to conduct a trial, it's required to prove the safety and the efficacy of a drug. So this was the first amendment. The Kafauver-Harris Amendment was the first amendment who provide the regulations that the safety and the efficacy of drug is needed to be there prior getting the approvals for that particular drug. So amendment was the response to the thalidomide tragedy. So the thalidomide tragedy which was happened. So due to this amendment was done. So it bills the present this particular bill was presented by the U.S. senators. So there was two U.S. senators who present this bill in the U.S. Uh, uh, Parliament. So Esther Kefauver and the Orrin Harris. So two U.S. senators which are there is the Esther Kefauver and Orrin Harris. So on their name itself, we have named this amendment as the Kefauver-Harris Drug Amendment. So so this amendment states which required a proof of efficacy required for the new drug approvals in addition to the safety. So which amendment was there for the safety and efficacy? So it was the Kefauver-Harris amendment. Then the thalidomide was not approved. So it was not approved. So after the passage of after having this particular amendment, it's required to have the NDA. So NDA is the, please tell me what is NDA? New drug application. So guys, you need to understand, like we have two things. We have the IND and we have the NDA. So IND is the investigational new drug and NDA is the new drug application. So we are filing INDs to initiate the clinical trials, and we are filing NDAs for marketing the drug into the for so that we can market the drug. So we are having the NDAs and we are having the uh, IND. So you need to understand the difference between the both. So IND is the permission to conduct the clinical trial and NDA is the permission to market the new drug. So you can understand from the name. IND is the investigational new drug. So whenever we are having the investigational new drug, we want to conduct the trial. So it's the permission to conduct the trials, and NDA is the new drug application. So we are filing for the new drug application, like this particular, we are having this particular drug and we want to market this particular drug. So whenever we are filing for this, so it's NDA is the permission to market the new drug. So after this, after this amendment was passed. So what are required? NDA needs. So whenever we are filing for the NDA, so NDA is new drug application. So up to NDA, how much phases we have done? Like for filing the NDA, what all up to where we have to complete the how much phases we need to complete for filing the NDA? We have already we have discussed this thing
We
yesterday.
have to finish animal studies
No,
and
guys, no, guys.
Ma'am, this is after phase 3, ma'am. We have
Yes,
to
yes,
apply for
yes, yes.
NDA
After
for
phase three.
marketing a new drug.
Yes, yes. Good, Anamika. Good. Yes. See, guys, see here. I don't want you to come confused. Like, see here. So, can you see my slides, Charita? Can you
Yes,
see?
ma'am.
So, like here.
yes, ma'am.
So, we are filing IND, that is the investigational new drug, to conduct the clinical studies. And after conducting phase 1, phase 2, and phase 3, we are filing for the NDA so that we can proceed further with the phase 4, so that we can market the drug and we can see the effects of a drug in the real world. We can see the effects of a drug in a real population for the longer duration of a time. So IND is filed for the clinical studies, for conduction of a clinical studies, and NDA is filed for the marketing the drug so that we can market the drug. So after this act, so after the Caffever-Harris Amendment Act, so for if we want to file the NDA, if we want to market the drug, so what all data is required? What Caffever-Harris Amendment says. So it says we require the data about the efficacy and safety. So for filing the NDA as per the Caffever-Harris Amendment Act, we require the data about the safety and the efficacy. If we want to file the NDA, we should have the data about the safety and efficacy. And when we are conducting the phase 2 and phase 3, so in phase 2 only we are getting the data about the safety and the efficacy, and in phase 3 we are going to confirm that particular data. So we are now having the data about the safety and efficacy, and we can now apply for the NDAs. So guys, are we clear up to here? I don't want you guys to confuse. So be clear up to here, then only we will proceed further, because these are the initial things, and I don't want anyone to get confused or having some kind of unclarity. So from the phase 1, we are having this data about the safety. Then from the phase 2, we are having the data about the efficacy and the safety. And in phase 3, we are confirming the data that we have gotten from the phase 1 and phase 2. That we are confirming the data about the safety and the efficacy. So when we are filing for the NDA, so we are having the data about the safety and efficacy. And this only the Caffever-Harris Amendment says. So if we want to file for the NDAs, we should have the data about the safety and the efficacy. So first point this was the after passage of the amendment, the first requirement was the need to show the safety and the efficacy data. Then the second requirement is the ICF. So whenever we are conducting the trial, so when it started from the phase 0, we are required to have the ICFs from the persons who are there in the trial. So ICF is the informed consent form. So informed consent form, you can understand it's a consent form. So we are making we are whenever we are having the patients in the trial, we are trying to make them understand these all things you are going to do. This is the procedure of the study. On this days you need to come to the site. This can be the side effects. So we have a proper consent form. We have a proper document where when if the patients agree to participate in the trial, then they are going to sign that consent form, and eventually the doctor also sign that form. So that consent form is signed by the patients, and after the signing the consent form, then only we can start with the trials. So this is the first mandatory step whenever we are starting the trials. We can't start with any procedure on the patients without having the informed consent form. Then this was the second thing. Then the third thing is the ADRs, that the adverse drug reactions are needed to be reported to the regulatory bodies or to the FDA, that is the Food and Drug Administration. So this was the Caffever-Harris Amendment Act. Okay. So continue with the establishment of the guidelines for the ethical trial conduct and the regulation for the drug development. More than 1019 novel drug therapies has been approved by the FDA, and that is virtually all medicines used today. So all the medicines that we are using today. Then we are having the thalidomide tragedy. So here you can see the drug was prescribed to the pregnant women for the Manning sickness. Instead, some babies are born to the internal deformities. Instead, the drug caused severe deformities in the babies with missing limbs. Missing limbs were seen, and it was estimated that over 3,500 babies died for their first birthday. Like 10 up to as per the data, like more than 10,000 babies were born with the deformities, and over 3,500 babies were died. So you can assume how like dangerous this drug was there. So guys, are we clear up to here? Like we are done with the most of the history. So now we are going to start with the types of a trial that we are having. So are we clear up to here?
Yes, ma'am.
Okay, guys. Okay, thank you. So the next thing that we are going to discuss is the types of clinical trials. So in clinical trials can be like it can be divided into clinical trials for and clinical trials can be. So clinical trials for is like it can be for prevention, screening, diagnosis, treatment, and quality of life. So prevention is like it involves like if we are going to find the better way to prevent. So it involves the healthy people and people who do not have any disease or have a high risk, do not have cancer or at a high risk of cancers. And basically, by having this prevention of trials, we look at we look at the cancer risks and a way to reduce those risks. I'm just giving you the examples. So in prevention trials, what happen? We are going to have two kinds of studies. So one is the action studies, you can say, and another one is the agent studies. So by having the prevention trials, we are looking the better way to prevent that particular disorder from developing or returning. So we have the action studies and we are having the agent studies. So in prevention trial, we have two kinds of studies: action studies and agent studies. So action studies is doing something. So whenever if the patient is doing something, so that this that particular indication, that particular disease can be prevented. Like in case of diabetes, the doctor asks the patient to walk, to manage your diet, to have a healthy diet. So this comes under the action. Like they are taking some action so that they can prevent those disease. They can prevent the reoccurrence of that particular disease. So in action studies, but things comes like doing something, exercising, eating healthy, all these things. Then we have the agent studies. So agent studies is like taking something. So when we take some medicines, when we take some vitamins, minerals, or dietary supplements, so that we can avoid that risk of developing of that particular disease, or we are finding some better way to prevent that disease. So that's come under the agent studies. So in prevention study, it's the better way. We are looking for the better way to prevent the disorder or from developing or returning back. And in prevention studies, we are going to have the two kinds of studies. So we are going to have the action studies and we are going to have the agent studies. Then the next thing is the screening. So screening is like prior having any something, we are going to screen that particular thing and we are going to assume like this thing can be happen and we can easily have the treatment for that. So it's aim to find out the best way to detect certain disorder or health conditions so that we can decrease the number of deaths. So like in screening, like we are screening the number of patients. So in corona, like I will give you the good example of corona. So when we are having the corona, so like after corona, you have seen whenever we are visiting some hospitals, like not hospitals, whenever we are visiting some markets, so there were we are having some testing. So random testing was done so that we can have the idea if someone is going to have the corona, if they are having the risk, so we can isolate them. So we can isolate them, we can provide the treatment. So that is good for them also, the person who is having the disease, and also good for the other population because they are not going to infect the others. So in screening, the goal is to find to the test the new ways to find the disease early, so that the we can find the disease early when it may be more easily treated. Because in initial days, like in cancer also, if we found the cancer in the early stages, so it's easy to treat that cancer, so that we can reduce the number of deaths which can be there from that particular disease, and we can reduce those number of deaths. So this is the screening trials. Then we have the diagnosis trials. So in diagnosis trials, we are going to have the diagnosis. We are going to have a refers to the practice of looking for a better ways to identify a particular trial. So in this we are going to have the diagnosis, and we are going to uh, see, so this particular disease is there, so it refers to the practice of looking for the better ways to identify a particular disorder or a condition. So by having the diagnostic procedures, we can identify, we can look the better ways to identify a particular disorder or a condition which the patient is having. Then the next one is the treatment. So in treatment, treatment, what happens? So they are providing that test new treatments or new ways of existing treatments. So sometimes we are already having that treatment. So we have the treatment for particular disease, but now we want to identify some new ways of treatment. Like we increase the dose, sometimes we reduce the dose, for the frequency for the dose, we increase the timing for the dose. Initially, the patient was taking the dose for the one time a day, but now we are going to, the patient is going to take that particular dose for the two times a day. So it can be like we are looking for the new treatment regimen or the new way for the existing treatment. So this particular treatment trials, it can be for the drugs, it can be for the vaccines, it can be for the surgeries or radiation therapy, or the combination of treatment. Then we have the quality of life. So quality of life trials is like they are the trials where we are going to find out the way so that we can improve the quality of life of the patients. So the patients who are having the chronic illness, especially those who have the side effects from the cancers or its treatment. So we are going to find out the better ways so that their quality of life can be improved. So in quality of life, we are going to have some different, we are going to approach some different methods, try to find out the new ways to cope up with the pain, nutrition problems, and might we test the drugs that can help, or we can have that drugs also that can may help for the treatment of that particular disease, or we can also add some activities like exercise can be added, healthy eating habits can be added, walking can be added, so that we can improve the whole quality of life of the patients who are already having the chronic disease. So this was the clinical trials for. Then we have the another type is like clinical trials can be. So in clinical trials can be, these are further divided into observational trials and the investigational trials. So observational trials, like as the name suggests, like in observation, we are going to only observe the things. We are not providing any intervention, like in routine medical care. So we are having the patients on the routine medical care, and on that, on those patients, we are not providing any different intervention to those. So they are having their own routine medical care, and from the symptoms, from the signs that we are having from those patients, we are having own kind of trial. We are having own kind of studies on the basis of the symptom signs that those particular patients are giving us, and we can have our study. So in observation trials, investigator assess the outcomes in a group of participants as per the protocol plan. So they have defined the protocol plan, like in OPD we are having 20 patients are there who are visiting for this particular disease, and we have divided those 20 patients into the two groups, and we are like on the basis of age, any factor, we have divided those groups, and we are going to see the effect of a drug on both these groups. So this is the own kind of observational trials. So patient will receive the intervention as per as per their or the procedures as per the routine medical care. So we are not going to provide any intervention as per the study protocol. They are only going to have their procedures and the medicines as per their routine medical care, and they are not assigned to any specific treatment. Like we are not going to assign to the placebo group or control group. So nothing is there, no control or treatment groups are there. We are only observing the outcomes in a group of patients or a group of participants that we are having. So this is the observational trials. Then we have the investigational trials. So what happens in the investigational trials, you can understand the core clinical trials that we are having. So we are having a product, we are having an investigational new drug, INDs. Investigational new drug is there for which we want to conduct the trial. So here we are going to divide the patients into the group. Then those patients will going to take the medicines, and this can be like group can be a control, or a case group can be a treatment group can be there, control group can be there, treatment group can be there, placebo group can be there, treatment group can be there, or the any other treatment which is already which is already approved in the market. We can have any different groups can be there, and we can compare those groups, and we can see the outcome for that particular drug on those groups. So we are going to compare. So we are providing the intervention. So investigational is like we are providing an intervention to that to those patients. So it can be for the drug supplements, it can be for the devices, for the procedures, and for the technique. So are we clear up to here? Like if anyone is having something, you can ask.
Ma'am, can you repeat the observational for clinical trials can be observational part?
Yes. Observational, like whenever we are having the patient in the routine medical care, so we are not providing any specific intervention to them. We are not assigning those patients into any groups like the groups that we are having in the clinical trials, in the randomized clinical trials, like we are having the control group, we are having the treatment group. So we are not assigning patients into any those kinds of groups. So we are like we are having the patients, like you can understand. So I am the doctor. I am having the like I am having like in my OPD we are having 50 patients, and for those 50 patients we are like we are providing like all those 50 patients are having diabetes, and they are taking the medicine. So like I have divided those patients on the basis of age, from the age of up to 40, from 18 to 40 there is a one group, and the above 40 we are having the another group. I have divided the patients on the basis of age, and we are providing them the treatment as per their routine medical care. And now we are seeing the effects of that treatment, about all factors like there, like whether they are having any side effects, whether the drug that they are having, like for some patients, like they are having the drug. So what all effects are there for the drug? So we are not provided any specific treatment. We have not restricted their ongoing treatments, and we are only observing the factors. We are only observing the outcomes that are there as per their routine medical care. So investigators assess the outcomes in a group of participants as per the protocol plan that they have decided for that particular observational trial. So patient will receive the interventions as per their or procedures as per their routine medical care. Or you can understand, like when we are in when we are in masters, or for the doctors who are going to perform their thesis. So they are not performing any clinical trials. They are only performing the observational studies. Like they are having the data in the database, and they are going to use, like they have decided their protocol, like what their protocol they want to see. Like you can take the example, like I want to see the effect of the pentaprozoles on the GERD patient. So I have designed my protocol. Then I will go and search in the OPD the list of patients which fall under my protocol. I'm going to take those patients in the protocol, and as per my protocol design, I'm going to divide those patients into the group, and will, like I'm having all the data of those patients in the software, in the electronic data, in the database that is there, like for medical records, but EHR, electronic health records. And on the basis of those data, I am going to provide my observation. So I am not providing any specific intervention to the patients. The routine medical care that the patients are having, only on that basis, as per my protocol, I am going to have the observations. So Namika, am I clear?
Yes, ma'am.
Okay, good to know. So anyone is having any doubt? Like I think like it is quite a new thing and also quite confusing also. But I want you guys to be clear at each and every step so that we can proceed further easily. Okay. So the next thing, these observational trials are further divided into the descriptive one and the analytical one, and the experimental trials are divided into the uncontrolled and the controlled. So let's understand the types in the observational trial. So in observational, we are having the descriptive and the analytical. So descriptive is like whenever we are describing the specific characteristics in the population of interest. So we are only defining the specific characteristics, like only the age or any specific characteristics. So this per descriptive trials are further divided into the case report and case series. So what is case report? So whenever the investigator discuss their experience with the patients on the basis of symptoms, signs, and diagnosis and treatment. So like they are discussing, they are going to discuss the their experience on the basis of the symptoms that they are observing on the patient, the signs, the diagnosis, and the treatment that the patient is having. So these are the case reports. Then we have the case series. So in case series, include, the investigator includes the patients with similar experiences, like the patients who are having similar experiences. If someone is facing the more headache, so I will put them in the same group. So these are the case series. So these kinds of studies are known as the case series. So in descriptive, we have two kinds. We have the case reports. We have the case series. So descriptives are particularly like these only describe the specific characteristics in the population of a interest. Then the analytical. So analytical is the observational studies without any intervention or the experiment. Purely these are purely on the basis of the observed outcomes. So in analytical, like we are going to have three further kind of studies. So first is the cross sectional, then other one is the cohort studies, and another one is the case control. So whenever we are designing the protocol of the observational studies, so we can use these designs to design our protocol. So what happens in the cross-sectional studies? So cross-sectional studies, like we are going to have the specific sample as a specific time. So like in this OPD, we are having the patients with the arthritis at a specific time at this particular time period. So we are going to take those patients at that time so that we can calculate the frequency of a disease. So what is the frequency of a disease in those patients which are there in that particular time? So by having the cross-sectional studies, we are going to calculate the prevalence, or you can say the frequency of a disease in that particular population, or you can say in the specific population and the specific time period. So these kinds of studies are easy to conduct, and like these can be easily conducted. Then the next thing that we are going to have the cohort studies. So in cohort studies, what happens? So cohort, we are going to have the cohort. So we are going to have the two samples. So one is for the risk and other is for the without risk. So groups are divided, like the patients are divided into two groups. So one group is that is having the risk, and other group that is having the who are not having the risk, so without risk. So by having this cohort studies, we are going to see the risk of developing, whether they are developing the risk or not. So like by having this cohort studies, we are also going to calculate the relative risk. So what will be the relative risk for the patient? So we have the patients, so you can understand, we have the patients who are having the tuberculosis. So one patient is we have the patients who are having the tuberculosis. So both patients are having the we are having the patients with tuberculosis, and one is the smoker and other is the non-smoker. So others are non-smoker. So we are going to see, so whether this smoking is going to affect, they already have the tuberculosis, but one group is smoker and other group is non-smoker. So we are going to see whether this smoking is going to affect or is going to lead to the lung cancers or not, or lung diseases. So this particular one group is with risk and other is without risk. So we are going to see the relative risk ratio for this population. So in cohort, this cohort is further divided into two parts, like prospective and retrospective. So prospective is like whenever we are seeing the things in the future flow, like in for the future things. So for the future prospective, and retrospective is like when we are looking in the past, the data that we are already having. On that basis, we are going to have our results. So that is the retrospective. Then the last thing that we are going to have in this particular is the case control. So in case control, what happens? We are going to have the groups. One is case and other is control. So case is the groups with the disease, and the control is the case without disease. So these kinds of, in case of cohort, we are having the retrospective and prospective both kind of studies. Whether in case of case control, we are only going to have the retrospective studies. So there is no prospective studies. So by having the case control studies, we are going to calculate the odds of having the risk factor or a exposure if an individual has the specific conditions. You can easily understand, like by having the case control studies, we are going to see the odd ratio. So what is the odd ratio? So odd ratio is the that particular population is having a risk or an exposure if an individual has the specific disease. So this is the odd ratio. So we are going to see the odd ratios from the case control group. So this is the observational studies. In observational and descriptive, we are going to have the case series and case report. And in analytical, we are going to have the cross-sectionals, cohort studies, and the case control studies. Then coming further with the experimental. So in experimental, we are having two groups. One is uncontrolled and other is the controlled. So uncontrolled is like they do not have any control group. So control group is like whenever we are going to have the control group is like the group who are not receiving the investigational, the group who are getting the placebo or not receiving the any investigational product. So in uncontrolled, we are only going to have the one group. So and that one group is only getting the investigational drug. So there is no placebo group or there is no control group. So this particular uncontrolled thing is performed in case of early phase. So whenever we are having the phase 1 or phase 2, so then we are using this uncontrolled. Moreover, it is only used in the phase 1, and phase 2 also we are having the controlled designs. So what we can see in uncontrolled group, so by having only the one group, we are only going to see the pharmacokinetic property of the drug, or the how the investigation like the investigational new drug is going to be tolerated in the body of the patients, and we can decide the maximum tolerated dose or the dose range for the drug. And we can also see the side effects, the tolerance, and the interaction or the efficacy of the drug. And then we have the control. So in control group, we are going to have the two groups. So one is the control group. So control group is like the group who's getting the placebo or ineffective treatment, or the other is the group which is getting the drug, or you can say the treatment. So it's basically common. So the control group is basically common in the phase 3. So we are having the control group in the phase 3. So placebo control can be there. No treatment control can be there. So no treatment control is like placebo control is whenever the patient is getting the placebo. And placebo is like the no active ingredient is there. So like what happens? Like placebo looks similar as the drug. So it looks similarly as the color in shape, in size, in quantity. It looks similar as the color, but it is not having the API, it is not having the active pharmaceutical ingredient in it. So it's the normal thing, it's only the salt which the drug is having. So the what can be the control group? So control group can be the placebo control. So whenever the patient is getting the placebo, it can be a no treatment control, like the patient is not getting any treatment, so no treatment at all. So then the other control group is the active treatment control. So like active treatment control is the new drug with a standard drug, or a comparing the combination of a new drug versus the standard alone. So like a new drug or a new drug with a standard drug, so the drug which is already there in the market, or comparing the combination of a new plus standard drug versus the standard alone. So we are having going to have the new drug plus the standard drug versus the standard drug alone. So we can see the effects. So by having this control group, we are going to evaluate or demonstrate the equivalence, non-inferiority, and the superiority of the treatment. And like this was the control group. So then the control group is further divided into the randomized and the non-randomized. So non-randomized is like we are going to have the two groups, but we are not going to have the random allocation. So what is random allocation? So whenever we are assigning the drugs to the group, so the process of assigning the drug to the group by some computerized method. So in a non-randomized trials, we are not having any such method. So we are assigning simply, and in randomized we are going to have the random allocation. So like a random allocation can be there. So like we are not aware, like that thing is different, like blinded and unblinded. So like we are using some method for assigning, like we are using some process for assigning the treatments to the different groups. So are we clear up to here, guys?
Yes.
So who runs the clinical trial? So doctors or researchers runs the clinical trial, and where? So at hospitals, at private specialties, at the research institutes, at the academic institutes, or other health providers. So these can be the places where the clinical trials are conducted. So in the next class, we are going to discuss this whole design so that you can have an overview how the clinical trial is conducted at the site, what all documents are needed. So like we are also going to have this specific class where I will going to let you know like when the trials are initiated, so what all documents are required for the initiation. So during the trial, what all documents are required. At the closeout, what all documents are required. So whenever the CRA is coming for the like the selection of the visits, so what is the ethics committee, so for the submissions, what all documents are required. So we are going to have those in depth also, but here I am tell you the flow, how the things are going on with the trials. Then we are going to discuss, this is nothing. So in the next class we are going to discuss this flow chart, and then we are going to start with the phases of clinical trial. And